back to Devans Biology. In this video series of respiration in plants, we have completed the metabolism of respiration. Now, after understanding the mechanism of respiration, in which we have completed 3 stages: glycolysis, pyruvic acid oxidation and Krebs cycle. Now, it becomes very important to understand the enzymes and functions of each stage. In the previous video, we had a look on enzymes and their functions that were used in glycolysis process. And today, let us have a look on the enzymes that were used in Krebs cycle. Enzymes and their functions. In Krebs cycle, the first enzyme that was being used was citrate synthase enzyme. Krebs cycle starts with the condensation of acetyl group of acetyl coenzyme A with oxaloacetic acid and water. This reaction was catalyzed by enzyme citrate synthase. So, the function of citrate synthase is condensation. In the next step, citric acid gets converted into cis-aconitic acid and then cis-aconitic acid gets converted into isocitrate. Both these reactions are catalyzed by the enzyme aconitase. The citric acid and isocitrate, these both are isomers. So, the function of aconitase is isomerization. In the next step, isocitrate gets converted into oxalosuccinic acid, and this reaction was catalyzed by isocitrate dehydrogenase, in which oxidation of isocitrate was done and NAD gets reduced to form NADH and H+. So, the function of isocitrate dehydrogenase was oxidation. Dehydrogenase enzyme catalyzes the reaction in which hydrogen atoms are removed instead of oxygen atoms in oxidation reduction reactions. Then this oxalosuccinic acid gets converted into alpha ketoglutaric acid. This reaction is catalyzed by enzyme decarboxylase. Decarboxylase means removal of carbon dioxide. So, in this reaction, there is a removal of carbon dioxide and the reaction was catalyzed by decarboxylase. So, the function of decarboxylase is decarboxylation, means removal of carbon dioxide. Alpha ketoglutarate then gets converted into succinyl coenzyme A. In that reaction, the enzyme used was alpha ketoglutarate dehydrogenase. As the enzyme used was dehydrogenase, so oxidation was done. The function of dehydrogenase enzyme is to oxidize the substrate. So, oxidation was done and alpha ketoglutaric acid gets converted into succinyl coenzyme A along with the decarboxylation reaction. So, in this also carbon dioxide molecule is being removed. After that, this succinyl coenzyme A is oxidized to succinic acid and the reaction was catalyzed by succinyl thiokinase. This was a substrate level phosphorylation. Substrate level phosphorylation means formation of ATP directly from the metabolites. Phosphorylation word means formation of ATP or synthesis of ATP. If it is a substrate level phosphorylation, then it is a synthesis of ATP directly from the metabolite. In this reaction, succinyl thiokinase enzyme is being used. And then in the next reaction of Krebs cycle, this succinic acid gets converted into fumaric acid, or succinate is converted to fumarate and FAD gets reduced to FADH2. In this reaction, the enzyme used was succinate dehydrogenase. Again, as you know, dehydrogenase enzyme catalyzes the reaction in which oxidation is being done. And then in the next step, fumaric acid gets converted into malic acid with the help of enzyme fumarase. What is the function of fumarase enzyme? Fumarase enzyme helps in hydration. Fumaric acid was hydrated to form malic acid. Fumarase actually catalyzes the reversible reaction, and in the last reaction, malic acid gets converted into oxaloacetic acid and the reaction was catalyzed by malate dehydrogenase. Now, as this is a dehydrogenase enzyme, so oxidation will be done. Oxidation of malic acid is done to form oxaloacetic acid. In all these enzymes, This enzyme succinate dehydrogenase, this is found in inner membrane of mitochondria, while rest all enzymes they are found in mitochondrial matrix. Enzymes are very important for catalyzing the mechanisms. So it's very important to understand, to memorize the enzymes and their functions. I hope this video will make easier to you to understand the functions of enzymes and it will be helpful to you. Thank you.